Good morning to everyone. I want to say thank you to the French colleagues that yesterday offered me an assist for this presentation. So today I present our work on geographic pattern of head morphology in Syngnathus typhle across European marine regions. So let me start with a question that many of you have faced in your work. So how do distinct morphologies persist across the no are because the geographic the answer is less clear because barriers are weak and gene flow is high. So Syngnathus typhle offers a rare opportunity to test this. It was a wide distribution, a limited dispersal, and important for our study, functional critical head morphology. Now, why focus on head shape specifically? In Syngnathus, we know that the head is not decorative, it's a feeding apparatus. So rapid suction feeding means that does not shape and jaw position directly determine the size and the prey capture. So elongated snout are better for targeting smaller and more evasive prey like copepods on the smaller amphipods, and shorter and deeper heads are associated with larger prey like decapods or fish larvae. So head morphology integrate ecology, integrate behavior and evolutionary history. Even subtle geographic variation in this structure carries a real ecological meaning. Given this, Syngnathus typhle is an ideal model species for this question. It's distributed from the Baltic to the Mediterranean, so 4 distinct biogeographic regions. Limited dispersal means that we expect a population structure, and most European species are sensitive, and morphological baseline are urgently needed. So, how did we approach to this? Our sampling covered 24 sites in 4 regions, and this was carried out as a part of Eurosync project. All individuals were adult and identified by minimum body size and secondary sexual characters. This was important to avoid any ontogenetic effect in our study. The protocol was completely non-invasive. All specimens were photographed alive. And this is important both ethically and practically, because it means that the protocol can easily repeated in the future for other species. We used a standardized lateral photography, and the landmark digitization was done with the TPS Dig2 software, and we put on the photograph 15 landmarks plus 3 semi-landmarks curves, giving us 195 points for individual. Measurement error was less than 3% of total shape variance, and the analytic pipeline is very, very long. So we had generalized procrusted analysis, then PCA, MANOVA, procrusted analysis, COA, disparity, and so and so. So what did we find? Regional differences are strong and significant. In fact, the regions explain 31% of total variance. The pairwise comparison show that the Atlantic versus Mediterranean is very significant, and also Atlantic versus Baltic is significant, but it is important that Baltic versus North Sea is not significant. These two populations are morphologically indistinguishable, so this is consistent with the shared post-glacial history. The CVA confirmed this pattern. In fact, the classification accuracy is 82% overall. This is mean that a specimen can be assigned to the correct region with 82% of based only on the shape morphology. The CV1, this one, yes, which captured the most of variants, describe the snout elongation and the mouth position. And the CV2? The CV2 captured the vertical head expansion and the cranial vault curvature. So the morphotype are clear. This one. So the morphotype are clear, and the Mediterranean specimen, the green one, have elongated and slender and flat profile, while Baltic and the North Sea, the red one and the yellow one, have a shorter head and deeper profile, and the Atlantic species, the blue, are intermediate. Then shape follows geography continuously. We ran a two blocks PLS analysis, shape versus geographic coordinate. And we found that 80% of shape variation is explained by geography. PLS1, this one, is negatively correlated to the latitude. So as you move on north, head shape change in a predictable way. The Mediterranean species cluster distinctly from all the other regions along the PLS1. And so the head shape doesn't jump directly between the regions, but it varies along the continuous latitudinal and longitudinal gradient. So this is consistent with the isolation by distance, the classical model of population structure driven by limited gene flow over geographic space. Then the Mediterranean is exceptionally diverse. So we calculate the morphological disparity using a Procrustes variance. And the Mediterranean has the highest disparity. This one. Yes, the Atlantic has the lowest disparity. So then the Melanobis distance tree show that Baltic has the greatest internal cohesion. So individuals within the Baltic are very similar to each other. And the Mediterranean show the highest divergence. So there is a lot of variation within the Mediterranean region. Several of the Mediterranean sites cluster more closely with the Atlantic site. Atlantic locality than with the other Mediterranean sites. So, and this is one. These three sites are more closely like this. This suggests habitat-driven convergence across geographic barriers. So sites with a similar ecological condition may have a similar morphology. So what drives this pattern? The first, the biogeographic discontinuities. The Mediterranean Atlantic split aligned with the Almerian-Oran front. And many marine species show genetical and morphological splits in these boundaries. The Baltic-North Sea similarity reflect their shared post-glacial colonization history, and this population likely share a recent common origins. Second, feeding ecology. The Mediterranean elongated snout is consistent with diet based on smaller and invasive prey, like copepods and amphipods. And the Atlantic and Baltic robust heads suggest diet including larger prey. This is supported by the dietary data on the literature. Third, the elevated phenotypic diversity is consistent with the ecological heterogeneity, but we cannot yet distinguish phenotype plasticity from local adaptation. So morphology could be responding plastically to local condition, or it could reflect the genetic adaptation. We don't know yet. So moving to the conservation implication, the Syngnathus typhle population are not morphologically interchangeable. So if these populations are evolutionarily distinct, they should be managed separately. So Mediterranean population deserve particularly conservation priority. This non-invasive protocol is directly applicable to other Syngnathus species without sacrifice animals. To conclude, three take-home messages. One is head shape in Syngnathus typhle is geographically structured across European marine regions, and the differences are strong, statistically robust, and biologically meaningful. Two, the Mediterranean is not just different, in exceptionally diverse, with elevated within region and trans-regional morphology affinity. And the third, the patterns align with biogeographic discontinuities and ecological gradient, but separating plasticity from adaptation requires genomics. Thank you.